Welcome, everybody. My name is André Lima. We are going to start the session in the Science Day that is called Environmental Science and Technology. Today, we have the opportunity to receive 5 speakers. Let me show some of them. The first one will be Flavia Braghiroli. She is from the University du Québec, ABITIBI. Then the second word is very difficult to say, but it's TÉMISCAMINGUE. I learned that is the name of the universities are from indigenous, right? Okay, she's from Canada, and she's going to tell us about her research on bioresources and carbon materials based on forest products for environmental applications. She will be followed by Aayushi Patel. She is from PENNSTATE, Harrisburg University at US, in Pennsylvania State. She's going to tell us about the product in vitro inhibitory effect of ramp derivative mixture and natural products on leukemia cells lines. And these are the two international presenters today, and will be followed by 3 speakers. They are from Univalle, our university. First of all, will be the Marcus Polet, is our coordinator of the graduate program in environmental science and technology, and he is going to tell us about this program. Followed by Professora Doutora Vivian Cionek, also from the Univalle. She is going to tell us about the stream aquatic biodiversity conservation, and will be the final one that I will say some words about our research on advanced marine genetics and biotechnology, what we have been doing here at Univalle. So to optimize our time, I will close my presentation here and ask Dr. Flavia to be opening her presentation. And while I will explain, or better, I will introduce Dr. Flavia for the people that are on this section. So now let me read some words here. Dr. Flavia Braghiroli is a distinguished researcher and educator in medical engineer and material science, with international research experience in Brazil, UK, France. She earned the PhD in chemistry from the University of Lorraine in 2014, and from 2016 to 2019, she was a Bating postdoc fellow at UKAT, that is a university on Quebec University, where she later coordinated the CTRI, Biomass, Bioenergy, and Bioproducts Laboratory. In 2022, Dr. Braghiroli became an associate professor at the University of Quebec, focusing on products and carbon-based materials for application of bioenergy, bioremediation, CO2 capture, wastewater treatment, catalysis, energy storage, and electrochemistry. So I would like to thank Dr. Flavia for to have the opportunity to receive here on our section, and it's a pleasure to listen to her presentation. I will close my microphone, but whenever you want something, just call us. We are around here, okay? Great. Thanks, Flavia, again for the opportunity.
Thank you. Thank you for the invitation. So today I'm going to talk about the subject that I've been working for the last 10 years. So bioresourced and carbon materials for environmental applications. So just a summary, I was at University of São Paulo, and then I did some studies in carbonaceous bio-based materials. Then I came to UCAT as a postdoc, and I worked with biochar made from forest residues for water treatment. And since 2022, so I've been working as a professor of forest bioproducts at the Institute of Research of Forest at UCAT. So UCAT is a very small university. It's placed in Quebec, and we have several campuses, but it's more or less on the northeast of Quebec. So it's a French-speaking university. I work in the laboratory of biomaterials and also for the 3D impression. We have other axes of research like ecosystem dynamics, biodiversity, ecosystem management, and forest product development and process. It's where I've been working with. So just to talk about a little bit how it works here, the research project. So we have several funding programs that support university companies partnerships. So it's the NSERC and the FRQNT. And then we have to have an industrial partner that are going to collaborate in cash or in kind. So just to let you know about the problematic is here. So in our region, Northwest ABITIBI-TÉMISCAMINGUE, so we have two main industries, is a forestry industry and mining industry. So basically these two companies, they generate a lot of waste. So what I try to do is basically use the waste from the forest industry to apply in mining industry. So it means transform wood residues into carbons, into adsorbents for water treatment and gas contamination, also for soil remediation. And so basically, this is my research program. I'm not going to talk about the results that I've been having since I started here. I'm just going to talk about the areas I'm working. So I'm very interested in any kind of this kind of waste, different kinds of conversion process, biochemistry, bioenergy, and the production of biomaterials. So basically these products, liquid and gases, biofuels, heat, biochar, bio-oil, and sungas. I'm very interested in any kind of thermal and thermochemical conversion process. Here in Quebec, we have some technologies that produce biochar, and one of them is called AREX Energy, which is a fast pyrolysis technology. So during my postdoc and also till now, we had some collaborations. It's like they have a pilot scale furnace, so we can produce biochar through this technology. And we try to develop other kinds of technologies to produce biochar. And then I also work with the application of these materials. And any kind of composite materials based on bioresource and carbon, resins, solvents, biopolymers, and bioplastics. There is also something that I'm interested in access to, is like the development of a forest residue conditioning center to improve biomass supply. So here we have a huge place and we have basically waste residue everywhere. So the idea is to develop a center that we could treat or condition biomass, and we could, and especially for certain applications, we could modify that biomass just to reduce transport, to facilitate the different actors from the forest for the forest. And also for the area 3, I work with the extraction of biopolymers, so cellulose, lignin, and tannin. I've got a project connected to the extraction of tannins from barks. So from the species from here, from ABITIBI-TÉMISCAMINGUE, we're like black spruce. Basically, we have other kind of species from the boreal forest. So I try to extract very biomolecules for produce renewable materials. And I also work with several kinds of applications. So especially the bioresource and carbon materials is a way of industrial waste management. We can use these materials in agriculture, carbon sequestration, degraded sites restoration. We are in a region where we have several mining sites that were abandoned. So it's a possibility to use these materials for for restoration of these sites. Bioenergy production, we can produce pellets made from wood or biochar and composites. There's a problem here, but no worry. So we can activate also these materials. So it means that the materials, they can have developed porosity and other characteristics that these materials could be used in energy storage, electrochemistry, gas absorption, effluent treatment, and catalysis. So basically, my objective is to look for partners that could contribute, collaborate with me. I produce materials, and then they are going to apply in different kinds of applications. So some of the projects that I have in progress, so development of porous bio-based filters to capture gases and particulate contaminants, production of bio-based materials from tannin extracted here in the region. We used 3D printing for the production of biosourced and carbon-based materials as CO2 sensors. And we have some projects that connected also natural sciences, engineering, and art. And so we are producing like objects made from 3D printing based on bioresourced materials. I worked with activated carbon, activated biochar for the application soils amendment. Production, characterization, application of functional materials derived from forest residues, and also the valorization of contaminated biomass. We have a big problematic here, which is the wood panels, end-of-life wood panels that we have a lot of this kind of materials deposited in outdoors, and this can provoke some contamination with the leachate. So this is a kind of subject that I'm interested in, and the pyrolysis of these materials is one of the solution for reducing these materials deposited in outdoors. Some of the group, the students that worked with me, I've been looking for students to work as in internship, master's, PhD. So if you have any students, we could talk about after. Since we had UCAT mission in Brazil, I think it was in April, we could develop some collaborations with Universidade Federal Fluminense, FATEC Sorocaba, USP, INPE, and I also have some collaborations with the universities in France, Université de Lorraine, Minales, Université Montpellier. So several partners, industrial partners that work in projects with me, and some of the partners that fund these projects, QUÉBEC, NSERC, and FRQNT. And we have here in Quebec and in Canada, we have a program for fellowships. It's called MITAX, and we can bring students for doing an internship during the graduation or during the master's or PhD. So this is a very good program that we could take advantage. And another thing that here I forgot to tell, that we don't have a graduation course in our institute. We just have master's and PhD students. So for this reason, we look for interns for coming here to work with research. So if there is, so I leave this, if there is someone interested, I leave my email. So thank you.
Thank you, Flavia, for your presentation. I put some notes around here. I don't know if we have time, but anyone that wants to make any question, we can have some minutes after each presentation. Flavia, you mentioned about the application of the residues from the forest at the application on mine. That's very interesting. We have some problem with mining areas around here on the south of our state. We could talk about that later. The biochar, we have professors that they are studying that as well. And we used to, I've been here for like 22 years, and about, I think 15 years ago, I have a friend that he was working with tanning, but using as a flocculant in wastewater treatment.
These as well.
I don't know if, great. And we saw the 3D printing is amazing, the idea of combining the science, technology, and arts using the research. That is very interesting as well. And it was great to understand that your institution have many opportunities for our students, and maybe also for us, the professor that are related to your activities or others. The MITAX program seems to be very interesting, and maybe is an opportunity to talk later with our students here. We can make some collaboration based on that.
Yeah, just a quick message that I need to work in collaboration with other people, because I produce the material, I characterize, so I look for other applications. So for me, this match could be very interesting. So the MITAX could be a good opportunity for starting collaborations with people from Brazil.
We have, I mentioned that we have simultaneous sessions right now, but also from the people at the pharmacy area, they are working with nanomaterials, and the people from the engineer courses that we have here, maybe they are will be interesting as well. So anyone have any questions? The ones that can speak, at least.
Or I can answer.
Andrea said, my friend, our friend here is a professor also. Andrea sent a question. Are there any governmental policy related to the use of natural-based products? Is this kind of product having any incentive?
I don't know if I understand the question. Let me think.
I think he is asking about the use of these material products. There's these residues. There are incentives to the companies from the government to use.
For sure, especially because, for example, mining companies or smelter companies we have here in Rwanda, so they need to heat at very high temperature. So we look for alternatives for, instead of using carbon materials, black carbon made from petrochemical sources, we could apply biomass instead. So for sure, now we have some politics here that aims to reduce for 50% of the CO2 emissions. So it's a lot. So for sure, the companies they will need to follow this. They will not have any choice. So they need to look for renewable alternatives. So for sure that they receive some how do you call money or some incentives for changing the consumption of petrochemical materials by resourced materials.
That's great to hear that, because we have the legislation that is asking for, you said 50% reduction, so it comes with incentive. And Marcus is helping me to check the questions. Is there anyone?
No, you can go ahead.
Okay. So if no more questions, we can go ahead with Aayushi Patel presentation. While she's going to put the presentation, I will try to make a resume of Aayushi Patel. Aayushi Patel, she is doing medical school. She has diverse experience in research, administration, and customer service. Currently, she is an administrator at Researchers for Change and previously served as a research intern at PENNSTATE, HARRISBURG in USA. Biofuel Laboratory for nearly 4 years. She coordinated patient experience at the intern U and Sheldon Medical Professional and Penn State Hershey Medical Center. Additionally, she worked at scheduling register at the Community Vaccination Center, retail attendant, and helped multiple summer research internship, showcasing a strong background in scientific research and practical healthcare applications. I'm sorry, Patel, I just made that so much fast.
It's all good.
Patel, she comes from the University of Pennsylvania, Penn State, Harrisburg, and she is going to make her presentation about in vitro inhibitor, effect on hemp derivated mixture and natural product from leukemia cell lines. Aayushi, I will close my microphone and you have your time. Thank you again for the opportunity to have you here. Thanks a lot.
Thank you. Can everyone see my screen? Okay, perfect.
Yes.
All right. So like I was introduced, I'm Aayushi. I'm a medical student at Penn State College of Medicine. Before I was at Penn State College of Medicine, I finished my undergraduate with Dr. Sairam, who unfortunately could not be here today, and he extends his apologies to everyone. He had to make an emergency travel and asked me to present it on his behalf. And I worked with him for nearly 5 years now, and as well as with our other collaborators, Dr. Desai and Dr. Dovat. So today I'll be talking about inhibitory effects in vitro of hemp, as well as hemp-derived mixtures and natural products on leukemia cell lines. Before I get into that, a little background. Nutraceutical formulations right now has a lot of market potential, especially when they are proven to, with data that it works through cell lines and animal studies. and that's for both like therapeutic perspective as well as the market perspective. Dietary supplements themselves have been known to grow to 8.8% between 2017 and 2022. Flax seeds, which is another supplement, as well as a therapeutic herbal supplement that can be used. Their market value was 528.8 million in 2020, and it has been expected to grow at a rate of 12.7 from 2019 to 2025, with the projected amount being 980. Moringa, which is another plant, is currently valued in 2018 at 5.5 billion, and that is also expected to grow. And then food application segment of turmeric was at 21.4 million in 2019, and it's expected to reach 52 million by 2027. Flax, we know, is a rich source for omega-3 fatty acids. A lot of omega fats is what flax seeds are known for, as well as other compounds that have bioactivity of value. Cell line studies have shown that flax seeds can improve cardiovascular health. The table over here also shows that cell lines have been used to show the effectiveness of flax on leukemia, as well as cancer and diabetes. It can also be used in combination with CBD, which is the therapeutic uses are anxiety and stress relief. For moringa is rich in proteins, vitamin A, minerals, and essential amino acids, antioxidants, and flavonoids. The extracts have been known for their nutraceutical and pharmacological functions, and it has a variety of properties, including anti-inflammatory, antioxidant, as well as blood lipid reducing functions. Both cell line and animal model studies have shown that moringa can be used to help treat diabetes, and cell line studies have also shown that moringa has anti-cancer effects on leukemia cells by inhibiting cancer cell growth. Moringa can also be used in combination with CBD for diabetes and leukemia as well. And then turmeric has oxidative and inflammatory condition. It helps with oxidative and inflammatory conditions or metabolic syndromes. There's a variety of benefits of turmeric that are currently being studied or have been elucidated through literature. And cell line and animal models have shown that active compounds in turmeric, curcumin, can actually help prevent and treat diabetes and its associated disorders. Cell line studies have also demonstrated that curcumin can effectively inhibit the growth of cancer cells. When turmeric is used in combination with CBD, it's again used for anxiety and stress relief. So my research specifically, as well as Dr. Sairam's research and our collaborators, was on medicinal effect of selenium on industrial hemp. This is something I closely worked with. Our lab over time has worked with other compounds and other combinations of natural products as well, which I will talk about later. But hemp itself, it's known for its secondary metabolites such as cannabinoids, and these compounds are what actually give the medicinal properties to hemp, and these compounds have been shown to show that hemp is effective against many diseases such as diabetes, epilepsy, pain management, and even COVID has been cited in literature. Selenium is a naturally occurring mineral with anti-cancer and antimicrobial properties, and selenium is used in chemotherapy right now in various cancers. It's part of the natural first-line treatment for certain cancers. Selenium has benefits and toxicity, and it's been more well studied in other plants than hemp, and there's very limited research with the effects of selenium with hemp. But our previous work showed that when we treated plants with selenium, our plants had a higher cannabinoid and total cannabinoid content, and the extracts from the selenium-treated plants were more effective against E. coli. So what we currently know from our current ongoing study is that we know that our plants are being able to uptake the selenium that we're giving from the soil, and we found that selenium in both the seeds as well as the flowers, and the flowers is usually where those secondary metabolites are found. We tested for genes within the pathway for producing the secondary metabolites, and they were all amplified with RT-PCR, and we're working on QRT right now. And then we also know that selenium plays a very important role in those pathways, especially the CBD synthesis pathway. Currently, what we're working on is doing cancer cell line testing for these extracts, as well as bioinformatic analysis of the effect of selenium on the plant itself. So the other ongoing study we have is just natural supplements in general and testing them on cancer cell lines, specifically leukemia. So the natural products used for this specific study was a curcumin, three different curcumin extracts and formulations, a flaxseed extract, CBC, CBN, and a hemp-derived mixture. Another formulation in here is a patented treatment, so that has been labeled as a patented treatment throughout the graphs that I'll show you later. The cell lines we used was MALT4 and NOM6. So for hemp-derived products on NOM6, we can see the IC50 graphs right here. So IC50 is the minimum concentration you need for a drug in question to inhibit 50% of the cells. So the lower the number is better because that means you need less of the drug at hand to treat a patient. So usually therapeutically, most drugs that are currently on, like medications that are on the market, they hover around the 12 to 13 mark for IC50s. Anything below that is really, really good. So as we can see over here, one of our combinations had a IC50 of 12.8, whereas the control, which is the current first-line treatment, is 1.0. Here, this one's a little bit better, which is the one of the curcumin extracts, and that was at 2.9. And then over here, when we did flax, our control was lower, but we also saw that a flax IC50 of 5.9, which is very good, especially if we know that current cancer treatments hover around 12 and 13. If we look over here with CBC, though, we can see that the control, what is on the market for treatment, was at 19.76, whereas for our product, it was at 12.64, which means it's more effective at a lower concentration. And here, as we can see, the IC50 was extremely low at just 0.7529. And then for here, as you can see, another low IC50 of 0.9. And then this one compared to the control, the other control that we had, where the IC50 for the control or the standard treatment was 57, the IC50 for our product was 9.3, which again highlights that we need even less to treat the same amount. And then down here as well, in this formulation, we can see that we had a lower IC50 again at a 1.173. And then for this hemp-derived product, the control IC50 was at 91, which is very high, but our formulation, and this is the patented formulation, was at a 0.6356, which is very potent, and you don't need a lot to make the same effect. And again, this is just highlighting the same thing where treated, which is always the red, is 0.3 and 2.1 respectively, whereas the controls were higher. And again, here, another hemp-derived mixture on NOM cells. Our treatment was at 0.45, whereas our control was at 2.9. And then on these graphs, we have CBC on our left here, and then CBN on our right. And again, treatment is lower for both, more so for CBN than CBC. And then CBN is also under that 11 to or 12 to 13 mark that they usually look for IC50s. And then we also found with flax seeds, our treatment flaxseed IC50 was 1.9, which is, again, considerably under the 12 to 13 mark. Great, thank you. Any questions?
Thank you, Aayushi, for your presentation. I was very impressed with the combination that you said. Using selenium, you can enhance a lot of the production on about the whole cannabinoids, and they are more effective. This is very interesting. And also those graphs that you showed to us, as you mentioned that the regular I see 50 is around the 12 to 14. You have showed many formulations that are very lower than that, and many about lower than 1. You showed one with garlic with 0.6. What's the next step? Is to try to patent, as you mentioned, on the, or there are many essays that need to be done before that?
So we are underway with our patenting process for both the selenium work. We have a provisional patent for that. The selenium work is also still ongoing. We're just still doing cancer cell line testing, and that's going to be included in the patent as well. Along with the other work that I showed, that is, we do have a formulation that is patented within that, and they are continuing work. After the cell line studies, the next step would be animal studies.
Animal studies.
Mm-hmm.
Usually you guys done studies with mouse and the small ones, right? Okay.
Yep, the small ones.
Flavia?
Yeah, I'm very interested in selenium. I'm not from this area of healthy, but I know that selenium is produced during the extraction of minerals here. Where does it come from? The selenium you use? Is it?
So our selenium that we used, it's pure grade selenium, so we don't get it from like a natural source.
Okay.
But the whole reason why we went down that route initially when we picked up this project and came up with this idea for the selenium half was previous work from our lab showed that hemp can be used for reclamation of mine lands. So we found that hemp can pick up heavy metals.
Okay.
So that was the whole reason why we decided that if we take selenium, which we already know has antioxidant properties, and we just give it as part of a water regime in their soil, so it's still like the plant is uptaking it, maybe it'll have like more beneficial effects. But there wasn't much studied for hemp and selenium for us to have like a more guided path, so it was more exploratory in the beginning.
The only problem that when we talk about metals in mining waste is because we don't have just selenium, we have everything together. But what I worked was producing biochar, absorbed selenium into biochar, and then use them as a soil amendment. So the plants could take selenium, and then this could be available for the cow.
Mm-hmm.
That's
very
because selenium
interesting.
is important. Yeah. So I'll get your email we could discuss after.
Oh yeah, definitely.
Thank you.
Thank you, Flavia. Also, we have a question on the chat and another one that a friend sent me, but he couldn't send on the chat. What are the chances of leukemia cells develop tolerance to this substance and not being affected anymore?
So that is currently being something that we're looking into. It depends on a lot of factors of developing resistance, but no matter what the treatment is right now in cancer, that is a big problem or a hurdle that cancer research is facing is cancer cells becoming resistant. So in addition to IC50, we're also working on getting the other number, which is the number before it causes too much harm to a patient, which is, again, a very important when you're thinking in the big realm of using it for therapy or pharmacological perspectives. So that number is also going to be helpful for us to know exactly what window do we have for treatment, because sometimes that resistance can be overcome with just going up higher, but there's only a limit as how much up higher we can go. But that is still ongoing, and it really depends on like the cell line or what type of cancer it is for development of resistance, as well as what you're using and how aggressive your cancer is, basically. So with our products, I don't have the answer to how long it will take them to develop resistance, since that's still kind of being under study right now.
Understood. And we have another question from Professor André, is what is unusual time from patent to a commercial product? How many years is a normal time scale?
So for a drug-related patent, which is the only patent I've ever been involved with, usually the patent time, the process can be, you submit your patent application that I have this discovery, and then you have to submit what you have as like why you think you have a discovery with it. And then you have 1 year to add more information or / provide more proof to them that you actually have a discovery at hand. And then after that, it all gets validated. And then depending on what it is, like cancer cell line testing or animal studies, they take years before it could be on the market for human use to begin with. With IRB as well, it takes 10 to 13 years is on the shorter end, usually. COVID made an exception when we had to find something quick. But that if we include that timeline in, it could take at least like 10, 13 years to get one patent fully approved for a drug-related patent. It could be shorter within like 2-3 years for something that is more of like a process or a technology-based patent. That doesn't necessarily mean you're giving someone a medication.
Thank you. And it's interesting. You said that we can submit the patent deposit and we have time to do more stuff and more investigation to understand better the effects and add this information to the proposal.
The existing patent, yeah.
Great. Andreas, okay. He also wrote, I was wondering if it was too different from Brazil. It seems to be similar. So it's similar. Great to know. I would say that it's faster over US, at least the deposit period, because here sometimes we can wait like for 7 to 8, 9 years to receive a return if it's approved or not. And during this time, we have to pay the annual fee. It's very interesting, the strategy here. So it's a discussion for later. Once again, I wish anyone any other questions? Sorry. Okay. I wish we would like again to thank you for the chance to have you around here and to present the research that you are conducting. And we are open for also collaboration with your group. Maybe we can talk later about that. We have some researchers here, not in the session, but in the university that are interested in some therapies and also most of them using phytotherapies. So it's very close to what they are looking for. So I think we can move to the third speaker today. Will be Dr. Marcus Poletti. Poletti told us that he couldn't turn on his camera. However, he can put his presentation. And before he starts presenting, I will make his introduction as well. I made with the others. He's going to talk about the graduate program in Environmental Science and Technology today. Marcus Poletti is a coordinator of this program, graduate program in Environmental Science and Technology at Univali. He's a renewed researcher and professor. He collaborates with the United Nations Pool of Experts for World Ocean Assessment and founded the Journal of Integrated Coastal Management for the Portuguese-speaking countries. His research focuses on integrated coastal management and regional and urban planning. Dr. Poletti has a postdoc in political science from the University of Santa Catarina, PhD and master in ecology and natural resources from the Federal University of São Carlos, and degrees in oceanography and geography from the Federal University of Rio Grande. He has specialized in coastal management at the top institution and has been a visiting professor at the university in Spain, Italy, many ones, and Uruguay. Thanks a lot, Dr. Marcos Poletti, for the opportunity to have you here and to present about our course in environmental science and technology. I will close my microphone, and it's your time right now. Thank you.
Okay, thank you very much, Andra. It's a pleasure to be here. And thank you very much, Flavia and Aayushi. It's also a pleasure to have you in our program and also at our university. And the idea is to exchange experience and also, of course, to connect our researchers and students. They are all here. We are very glad for this day and this opportunity. Well, I'll talk about the graduate program in environmental science and technology. Our program is related with the Sustainable Development Goals, almost all of them we are trying to. And the postgraduate program trains human resources to work in the areas of teaching, research, and development through knowledge and mastery of tools and technologies aimed at the environment. Well, we have during the last 25 years, almost 25 years, we have already had the opportunity to form 280, almost 280 students of masters and PhD. And just to understand where we are located, we are in Brazil. We are located in the state of Santa Catarina. It's a state in South Brazil. And in Santa Catarina, we are in the city of Itajaí, which is about 100 kilometers from Florianópolis, the capital of the state. And it's a very nice area. It's an estuary area with where we have the biggest river in the state. This area, it's very important, especially in terms of civil construction, real estate, tourism, industrial and artisanal fisheries, ports and terminals, and also shipyards. It's a very important area today in Brazil. The University of Itajaí today is the first, and it was the biggest communitary and private university in Santa Catarina. We have about 24,000 students, and we have 7 campi, which is distributed especially in the coastal area of Santa Catarina. And our program of science and technology, environmental science and technology, is located in Itajaí. It's a very nice place for people to study, to make research. And our research lines are related to technology for environmental management and sustainable cities. And we have 2 different lines, and also in aquatic ecosystems, where we also have 2 different lines of study. In 2025, we are going to change one of these lines of research, and we are now, and this line tends to be about vulnerability and adaptation to climate change. So our team of researchers is, we have from many different fields of knowledge. And today we have 12 permanent teachers and 5 collaborators. And also we have 1 teacher from the University of Alicante in Spain, and also 2 students from postdoc. We have also about 80 students from many places and fields of knowledge. And our infrastructure is related to 16 laboratories, which almost of them have the researchers as the responsible of the laboratories. We also have 3 different magazines that are related to our course. One is called Integrated Coastal Zone, which is a magazine of coastal zone management. Today, the support is related with the University of Oporto in Portugal. We also have the Ecotoxicology, Environmental Contamination. It's another journal. It's very important in terms of ecotoxicology in Brazil. And also the journal of our course is the Brazilian Journal of Aquatic Science and Technology, which you are invited to publish in it. Okay. Well, our postgraduate program is the students and the professors. They are all involved in many researches, and also the idea is to put the course for the community, and especially the community around the university. And since then, we have been worked in some symposiums in order to show our different researches. Many of our researchers have got some prizes as their researchers in several areas. Our classes are our classes. Generally, we put the students on the field, which is very important to have knowledge, especially with what's happening in the field and in the area we live in. We have many extension projects, especially with local communities. Also, projects involved researchers. These projects, they are related especially to environmental issues. And many of these projects are linked with the private sectors. And many of them are very important, as, for example, the PMP, which is a monitoring projects on beaches, which is today perhaps one of the most important monitoring projects in coastal areas in the world. It's very important. And we also have many projects with shipyards and ports, and also governments around, international projects related with fisheries, which is one of the fields we have been studied during this period of time. And 4 years ago, we began also a project with the university, one university in Maranhão. It's a Brazilian state in the north part of the country, in the Amazonian area. And we involved 14 students. They are finishing this year their PhD. And also in this opportunity, we are launched an observatory of the Oriental Amazonian. It's the first observatory in this area. And the idea is to engage these 14 students to continue their studies. But of course, in terms of research, we think it's a very important project for the next years. And also, as you are here, Flavia and Aayushi, the idea, of course, is to exchange our experiences, especially with abroad universities. And we invite you to be here, of course, in Brazil, in order to make it happen. But we are also trying to make some relationship between other universities, as for example, University of Alicante in Spain, also the South University of Peru in Lima. Professor André also began some projects with the University of Alicante and also Perugia. Professor Vivian, for example, as she's here also in Australia. And this I suppose that this is the idea. The idea is to make things happen, to make this opportunity of this day to continue in our exchange, and making and the possibility also to move students and also our researchers to your university as you come to Brazil, and we are pleased to receive you here. Thank you very much.
Thank you, Marcus, for your presentation. It was great to show what we are doing here and the quality of the research and the way that we do the stuff here at our course on scientific, environmental science and technology. And maybe Flavia and Aayushi, maybe they can figure out a way to collaborate with us and or other researchers from their university as well. Any questions for Marcus?
For sure. I think I met someone from your university in São Paulo when we had a UCAT mission, but I don't remember now the names, but I'd be very interested and open for collaborations with you, for sure.
That's great. That's great.
Okay.
As you said, you have some programs at your university that is easier to
Exactly.
establish and start something together. And
Okay.
I would love to go to Canada.
For sure, we welcome students because we are a small university, so the students here, they are very welcome, and for sure. And I'm bringing now some Brazilians here, so we are gonna receive 4 soon. So for sure, if you want, we can have a meeting afterwards for discuss about that.
And Flavia, I would like to know, your university is a coastal university or?
Do you mean if you pay for studying? Is it
no, in terms of coastal area, it's located near the coast?
No, it's not near the coast. It's very northeast. Yeah, not near the coast, but.
What is the province?
It's Quebec.
Quebec, okay.
But we are not on the coast. Okay. We have one campus that is in an island that could be interesting, maybe for Vivian. Is a île, 7 îles. It's an island that so we study everything related to forest there.
Fantastic. Thank you, Flavia.
Okay, thank you.
And we received a question from the chat. Is from Desai Demand. Maybe he's from the group of Aayushi, I'm not sure. And is asking if besides oceanography, we do some studies with natural products and cancer. We do. My group we are going to present at the end of this session, but also, as I mentioned, the people from pharmacy, I'm not sure if is there anyone on this section, but they also do some study with natural products. We do with bacterial products and microbial and anti-cancer as well. So can we move on? Well, I would like to introduce and also thanks for the opportunity to have her here. My colleague, her name is Vivian Mello. She's a professor at our course on environmental science and technology. While Vivian is putting her presentation, I will present as I made for the others. Vivian holds a biology bachelor degree and PhD in freshwater ecology. Her work focuses on stream ecosystem functioning, aquatic community ecology, response to anthropogenic impacts and climate change, and biodiversity conservation. Vivian is the current Deputy Director of the Tropical Water Research Alliance in Santa Catarina, and a member of the Working Group on Freshwater Conservation of the Brazilian Limnology Association. Once again, thanks for the opportunity to have you here, Vivian, and I will close my microphone and...
Okay, thank you. You can see my presentation? Okay, nice. Well, good morning to you all. It is a great pleasure to be here in this scientific meeting with you. We'll be shifting a little bit of research focus. Now I will be talking a little bit about freshwater ecology. As Professor André mentioned, I am a biologist. So I am mostly interested in working with stream aquatic biodiversity conservation. So I have been a faculty member of the graduate program in environmental science and technology for 1 year and a half. I actually did a postdoc in the program starting in 2020. So now I am a faculty member, and this year I began working in the laboratory of community ecology. So a little bit about my research. Over the last decade, I have been working mostly aiming at advancing ecological concepts, but also contributing with the, also contributing with the production and development of management plans for protected areas in Brazil, developing assessment tools using biodiversity as indicators, and also evaluating ecological ecosystem process to inform about how is the magnitude or the effect size of anthropogenic action over small streams, but also using this kind of ecological approach to establish some kind of monitoring of a very complex ecosystem process to help us assist us with the ongoing restoration programs. So I did see a little bit of possibility for us to collaborate with Flavia on this, to work with projects that might assist with the stream restoration within our watersheds. Also, more recently, I have been developing more interested in evaluating mechanisms that explain how energy transfers from the terrestrial environment into the aquatic environment, because within watersheds, this is a very important process for us. So evaluating how it is happening is also very important for us to understand how the watershed is behaving, after all. And also working with how biodiversity provides us with ecosystem services, especially in river floodplains ecosystems and also coastal lagoons. I'm also interested in working with beta diversity. I have developed during my postdoc in Univali. I started working with ecological uniqueness, beta diversity in coastal shallow areas, and also looking for better strategies for us to evaluate how forest, the riparian forest, should be evaluated or restored under what kind of configuration under the landscape, so we get the better results, so we get better biodiversity results. Because there is an understanding in my line of work that the taxonomic, functional, and more recently, the genetic diversity of aquatic communities are very important to maintain the functioning of the systems. And I am aware that most of our population do not value biodiversity itself, but since biodiversity promotes and function and helps with ecosystem process, we are working with some kind of relationship of the importance of the maintenance of biodiversity into the provision of water and recreation for our systems. Oh, also important to mention, one of the latest contribution in which I participated, it was the production of this paper within all of the professors from our program. We were trying to give some directioning about how to generate scientific knowledge or how to direct our research to work mainly with climate change. That is a new line, as Professor Poletti mentioned in our program. It is also a new direction of research for me and for my research team. So we are actually now mostly interested on how connectivity on the watersheds are prejudicial or influential over ecosystem processes and the distribution of fish and invertebrates along the watersheds, mostly because this is the Santa Catarina, the state where we are now. You can see that we have a very large mountain range very close to the ocean. So all of these light green area are the places where we have our watersheds, and they are very small and they are very unconnected because they do not connect over land, and they have an environmental filter, that is the salinity. So we do have populations that do not exchange easily over time. And because we are under an area with a lot of people, a lot of population density and urban development that is very fast, we are seeing our watershed being even more disconnected. So for our latitude, we have the expectation of an increase in rain and extreme events in our region. We are already facing it. It's becoming even worse with time. And one of the effects of this flashy rain and these spates over streams is that it carries organisms, fish and invertebrates, but also it displaces and and changes the physical habitat of the streams. So when fish population try to recolonize the systems, they end up finding dams that are used for water retraction or energy production, pipelines, or even pollution as an environmental filter. So when we connect both of these pressures that are very, very important for us here, we can try to address research questions on where can we find hotspots of biodiversity conservation within fresh waters, so we can direct some conservation efforts and maintain our ecosystem services provision in our ongoing research and potential collaboration opportunities goes from the research on coastal stream fish, the Capoda and bivalve beta diversity and their susceptibility to climate change. Now, I am collaborating with Professor André Lima, as I mentioned in the beginning of the section to Flavia. For today, Roger is starting his postdoc with us. We are going to investigate how is the genetic diversity of fish population that are under extinction risks. We do have our red list of vulnerable species, but we do not know where they are. We are also working on the impacts of sand mining in some of our watersheds, some of the conflicts that we are facing here more regionally, and the susceptibility to climate change also. I do have some work within the Tropical Water Research Alliance. We are studying biodiversity gaps and the importance of Brazilian environmental legal protection for fresh waters. And the alliance itself has over 200 researchers, but on this specific work, we are working with some professors from University of Paraná, UNB, it's Brasilia, a lot of states from Brazil, and also from Australia. And the last one are the evaluation of small hydropower plants. We do have a lot of those here in Brazil and a lot of expansion plans in Santa Catarina specifically. And we do have an understanding that rivers should flow one way around downstream. And although we need energy, we are placing too much barriers along our watersheds and disrupting connectivity. We are changing the systems in a way it cannot be resilient, it cannot recover from pressures, and we do have to face climate change alterations that are mostly related to water here in our region, and erosion and silting, everything like that. So this is the main focus of my research here. And I really appreciate the time. It's a small presentation. I know I am still on a very initial phase of our work. We are still trying to spatialize work and discover a little bit of our streams here. And I'm open to questions and to talk to you about potential collaboration. I really appreciate it.
Thank you, Vivian, for your presentation. The idea was to have a different aspect what we have been doing in the environmental science and technology. And now with presentation of Vivian, we have an idea of the ecological point of view. So I will present some aspect on the technological point of view, both related to the environment. And when we think about the importance of the other two presentations from our friends abroad, it's interesting because we can find some relation from the aspect of the using of the byproducts in a way that they are not going to contaminate the rivers. And so you have this kind of connection. And also we can think about the preservation of this area, and the conservation of biodiversity is a way to have time to explore all the biodiversity available for the application on medicine on other areas. So we have to consider the ecological and preservation of environmental a very important area that is related to all we have been feeling on the last year with climate change, and is all related to what we have been doing in our group. Thanks a lot. It's a great opportunity to have you around and start collaborating with your group as well. Do we have any question? I saw that Flavia, Vivian here is a professor that works with subjects, so just click on the link.
I just click it and I will take a look.
Great.
It's nice. Thank you, Flavia.
I think I will move for my presentation. I would like to introduce, then I click here, just a little bit. So I'm a researcher and a professor at the University of Vale do Itajaí with a degree in biological science as Vivian at Unesp, and a master PhD in genetics at Zalc USP. I conclude the sandwich doctorate at Rutgers University and a postdoc at research at Jamstack in Japan. In 2002, I get into Univali, and I teach on the undergraduate and postgraduate programs of biology and environmental science and technology. And I lead the group that work with genomic and metagenomic data, bioprospecting of marine bacteria, and developing genomic tools for studying marine animals and phylogenetic and population. I have already advised more than 60 undergrad students and more than 30 postgraduate students, and have some papers around. So let me put my presentation now. Here is a very short one. The idea was to just show what we have been doing in our group. It's on? It's okay? Great.
Yes.
So the idea is to talk about advancing marine genetics and biotechnology, the key research activities that we are doing in our group here at Univali, with collaboration with many other professors from this university, from Univali, and also from other institutions and universities in Brazil and abroad. We designed this picture last year in a way to try to translate what we have been doing or we'd like to do in the next few months or years in our group. Most of what we have been doing is related to this section here, is the evaluation of bacteria, deep sea bacteria mainly, for their biotechnological potential. As I said before, we are looking for genes, enzymes, polymers, and also products that have some activity inhibiting bacteria, fungi, and also related to cancer. I will show not some results, but how we work on these 4 sectors. The second one that I would like to talk about is the biological inventory that we are creating here with our partners. As I said, the professors here from Univali, they are very active in the studying a series of animals from marine ecosystem, and we are trying to give some input on genetic analysis of those animals. And after identifying these organisms, we are trying to develop some strategies to characterize their diversity through studies of phylogenetics and population. Related to all these aspects, we are using now eDNA or environmental DNA to understand or to monitor environmental ecosystems for their activities, or related to what the information from the bacteria can reveal about the behavior of some animals. Let's see some more slides to understand all these 4 sectors. So from the biotechnological point of view, we have some strategies that are mainly the usual ones. After isolating the bacteria, we start evaluating their characteristics, and if they are interesting for any product that I mentioned, enzymes, polymers, antimicrobial, pigments, we sequence their genome, find their genes, and maybe we express these genes in other hosts. But besides the strategy of cultivating the bacteria, we also have the opportunity to develop the strategy of prospecting environmental DNA. I mean, the DNA from the bacteria, but from organisms that are not cultivated in the laboratory. Basically, we extract the DNA, sequence it, look for enzymes that we are interested in, clone them, and then express in a host. So we extract the information from the DNA that we collect on the environmental, and now we also are applying some synthetic biology, and we are using DNA from the data banks that are available for the public. As a result of this project, we studied the pathway to produce some carotenoids in this bacteria, Erythrobacter. We have some collaboration with the people from Japan, where we characterize some compounds with fungi anti-activity. Also, the pressure on the microbiome associated to some samples on the environment, the genome from bacteria. So here is just to have an idea what we have been doing related to the biotechnological potential of bacteria, deep sea bacteria. But on the last couple of years, we have the chance to implement in our lab sequencing platform. Is the sequencing platform called Mini-IO, is a next generation sequencing. And what we did a little bit different with this platform is to establish a way to sequence many kind of samples in parallel. So this could allow us to implement other strategies of research in our group, such as to work with the biological inventory. I mean, to study, to identify animals that are being studied by our collaborators, but they need to be identified. So I just put some pictures here. Some of the animals are very common, but others are quite unique. And having this chance to increase the quality of the research using the molecular identification, it was a great pleasure to us and opportunity to collaborate more with our other colleagues. So the idea here is to identify organisms that come from marine ecosystem in a regular way. You sequencing the COI gene, is a cytochrome oxidase 1 gene, and using that platform, the Mini-IO platform, to do that. So I can sequence and identify 50 organisms at the same time. This is good to scale up our capacity. Besides, of course, identifying the animals, we already had some expertise in studying the bacteria. So here are some of the papers that we published related to identification, to study of a diversity of bacteria, isolation of other organisms that are also important to understand the biodiversity and metagenomics on the marine ecosystem. And looking at the spectrum, or that section that I mentioned, diversity and characterization, we are also extracting more information from the platform Mini-IO. We decided to try to sequence the whole genome of mitochondrial genome of some animals. We are starting with this Tunus macaque. Tunus macaque is one of the most important tuna, is the bluefin. It has a very important relevance to the economy, and the sequence of the complete genome of an animal we call the mitogenome just to have an idea, this Tunus McCoy just have 2 sequence at the NCBI, so at the data bank. We are increasing, adding a third one. And this could be done with some techniques that we develop here at our group. And using the mitogenome, we can infer about the phylogenetics of these animals, and also use this information for population studies. So we are doing this first approach with tuna and also with some birds, the albatross. Looking at the aspect of eDNA for environmental monitoring, we are conducting some projects here with our students. This is one of the projects from Erica. We have others. And she is looking to use the eDNA to understand anoxic areas that we already detected with at our area here at Itajaí. This is a project also with collaboration with another project, Dr. Jurandir. And besides looking the DNA at the water, you also looking the DNA, the environmental DNA associated with animals, with the microbiome from albatross. Here is a project from our PhD student, Annie, and also from squid microbiome. This is a project with a collaboration with another university, and is being conducted by our students from Perugia University as well. So understanding the microbiome associated with the as animals, we want to go forward in understanding how about their diet, and how the treatment with this animal can affect their community or their health. So as a final considerations, we may say that understanding the biodiversity of animals, microorganisms, and associated eDNA can be done using this strategy that I mentioned, the techniques using the the genetic of identification, sequencing mitogenomes, and so on. The mitogenome, as I mentioned, can be used for phylogenetics and population studies. And of course, we already showed, and many projects and researchers are doing that, the potential of bacteria for application on biotechnology. We have received some grants from different institutions, nationally and abroad, and we have many collaborators from here and also from other universities. Here, the main collaborators that we are working together, many are from here, Univali, but also some from abroad. And of course, we need students. They are the good part of our activities at the laboratory. And I'd like to invite everyone to visit Itajaí, especially Flavia and Aayushi that are listening. These are secret spots inside the center of the city. You can imagine how beautiful is here, and you are all invited to visit us and to start working together. Thanks a lot for your time, and I'm open for any question. We are right on time, 12:00, as I would like to do. Any question?
There is a question in the chat, André.
Any interest in natural products from aqueous pellets like algae? Yes, our group didn't work with this material, but I'm not sure. But I think the people from pharmacy is already working with freshwater algae. I can put you guys together. Thanks a lot, Aayushi and Flavia, for your consideration about the meeting. It's a short time, of course, but it's a way to put the people that are far away together, at least for a few minutes.
Thank you very much for the invitation.
Thank you. So if we don't have any question, I'd like again to thank all the presenters, the speakers, and all the people that are present at our section. It was a pleasure to be here, and we will follow with the presential way. Unfortunately, Flavia and Aayushi won't be here, but we have some drafts to drink and some more presentation during this afternoon. Our Science Day is very, very interesting here, and we love it. The problem is that we just have one each year, but it's enough. Thanks a lot, people. Bye-bye.